At the moment, model organisms are widely used to study various properties and processes of living nature with the help of laboratory experiments. Model organisms is a non-human species that is extensively studied to understand particular biological phenomena with the expectation that discoveries made in the organism model will provide insight into the workings of the other organisms. There are different types of model organisms, including genetic, genomic, and experimental. Genetic model organisms are species amenable to genetic analysis. In other words, species that can be easily used for genetic research. Genomic model organisms occupy a special position in evolution or have a particular genomic quality which can be used for reference. Experimental model organisms may not be genetically amenable, but have experimental advantages, such as particular characteristics specific to the experiment.
In most cases, the criteria for selecting these types of model organisms are similar, and they include the following characteristics: producing large number of offspring, having a short lifespan, developing to maturity rapidly, easy to grow and maintain in restricted conditions, having a sequenced genome, and well understood growth and development.
Hamsters meet most of these criteria, therefore they are one of the most popular model organisms. Hamsters are representatives of order Rodentia, belonging to subfamily Chrysalidae, which consist of 7 genera and about 20 species. They live mainly in open arid landscapes, forest steppes, semi-deserts, and deserts. However, some species can be kept as house pets or laboratory animals. For example, Syrian or golden hamster, Chinese hamster, Campbell's dwarf hamster, and European hamster. It's worth to note that Syrian and Chinese hamsters are fiercely territorial and should be housed sanely. However, Campbell hamsters are social and live in a group. Hamsters are small to large muroid rodents with compact bodies, small furry ears, short legs, wide feet, and short stubby tails. Color vary among species. They can be gray, pinkish buff, light brown or reddish brown on the dorsal surface, and white, gray, or black on the ventral surface. Their flanks are often white as well. Some have a mid-dorsal stripe. The hamster stomach is divided into 2 distinct areas: glandular stomach and non-glandular forestomach. Their hearts are 4-chambered. The pulmonary and aortic valves are tricuspid. The respiratory system of hamsters consists of nasal cavity, nasal sinuses, pharynx, larynx, trachea, and lungs. The average total weight of the hamster brain is 1.4 grams. The cerebral hemispheres consist of large frontal, lateral, and occipital lobes. Breeding onset is at approximately 90 days in both females and males. Also, females may have their first estrus at 35 days. Hamsters are polyestrous and breed year-round. Ovulation is spontaneous. Duration of the estrous cycle is 4 days. The average gestation period is 16 days. Litter size vary from 5 to 9. The young weigh 2-3 grams at birth. Newborn hamsters are hairless and have closed eyelids and ears. They are weaned at 21 days. Cycling resumes within 18 days after weaning. This means that the litters intervals can occur every 35 to 40 days. Pseudopregnancy of 7-13 days duration occasionally occur as a result of infertile matings or crowding of females. Cannibalism is more common with hamsters than other laboratory rodents, and is usually associated with primiparous females during the first week postpartum. Other factors contributing to cannibalism are lack of nesting material and disrupting the female and her litter. For these reasons, the female and her litter should be left undisrupted for the first week postpartum.
In laboratory practice, several types of hamsters are used, but most often the Syrian hamster. This hamster is often used in experiments instead of mice, rats, and guinea pigs. This is an unpretentious, hardy, and prolific rodent that does not hibernate. Unlike rats and mice, hamsters need less nutrients, although they need more fat in their feed and also consume more food. Hamsters are used to reproduce various infectious diseases, such as tuberculosis and poliomyelitis, as well as in various research in the field of oncology and pharmacology. Entamoeba histolytica is a species of parasitic protozoan, such as amoebozoa. It causes a serious illness, amoebiasis, amoebic dysentery, amoebic colitis, which has such symptoms as fatigue, fever, and chills, abdominal pain, diarrhea, which can contain blood, mucus, or pus, intermittent constipation, and also nausea and vomiting. Syrian hamster can be successfully infected with Entamoeba histolytica. Similar to symptoms after human infection, extraintestinal complication is also found in the hamster. In the hamster, liver recruitment of neutrophils is the initial host response to Entamoeba histolytica infection. A study indicated that leukocytes can induce Entamoeba histolytica trophozoites to undergo cell death. Although an antiparasitic drug exists, side effects of toxicity exist in patients. Thus, this model has been used to develop alternative therapeutic agents. Throughout the period of the coronavirus pandemic, scientists around the world worked to create a vaccine, and on June 19, 2020, researchers from the Israel Institute for Biological Research published an article stating that a single dose of the coronavirus vaccine is effective against this disease in Syrian golden hamsters. According to scientists, their vaccine leads to the rapid and powerful induction of neutralizing antibodies against SARS-CoV-2. During the research, they used two groups of hamsters. One group was given the vaccine before being infected with the coronavirus, and the other was not given the vaccine. As a result, it was found that hamsters receiving the potential vaccine did not lose as much body weight as unvaccinated hamsters. The rodents from the first group also showed only mild lung pathology and no viral load, while unvaccinated hamsters suffered from extensive lung tissue damage and exhibited high viral load. In this way, the researchers believe that their vaccine is safe and effective enough to fight the coronavirus infection. Syrian hamsters are also a model for research in non-small cell lung carcinoma, which is one of the types of human lung cancer. In research, when hamsters are injected with a carcinogen several times over 6 months, they will develop that sort of cancer. In both Syrian hamsters and humans, this cancer is associated with mutations to the specific gene. For various reasons, collecting data on the way that Syrian hamsters develop this lung cancer provides insight on how humans develop it.
Syrian hamsters are used in study of infections of Leptospira interrogans. Pathogenic Leptospira interrogans is spirochete bacteria responsible for leptospirosis, a blood infection. Signs and symptoms can range from headaches, muscle pains, and fevers to bleeding in the lungs or meningitis. Syrian hamsters has been used to test the efficacy of vaccines against this disease. Syrian hamsters is the preferred model to study the infection of serovirus of Leptospira interrogans, with bacteria traveling rapidly to the bloodstream via the lymphatics, then invading tissues and infecting all organs prior to the death of the hamsters. Zoologist William Russell and microbiologist Rex Birch developed the 3R principle in 1959. The 3R principle is generally accepted standard that has given rise to the desire to search for and create alternatives, and thereby significantly reduce the number of laboratory animals used. Refinement – improvement, which means humanization in the preparation and conduct of the experiment, from the beginning of the animals keeping in the laboratory until their death, using non-traumatic methods and painkillers. Replacement is the variety of replacement technologies, when you need to replace animals with other models and techniques. Reduction – reducing the number of animals. If it is not possible to replace the animals in the experiments with other models, you must try to build the experiment in such a way that the minimum number of animals is used. Methods that can be used for research instead of animal experiments: models, mannequins, and mechanical simulators, computer simulators and virtual reality systems, experiments on plants, in vitro techniques on cell culture, use of dead animals obtained from humane sources, for example, animals that died of natural causes or were humanely killed after scientific experiments, and etc.